All right, welcome back. Today we're going to just finish up the nucleic acids. We already did the other three organic molecules of the body. We did carbohydrates, we did lipids, which are fats, and we did protein. But the fourth type of organic molecule in your body are nucleic acids. And just like proteins and carbohydrates, they have monomers, they have long carbon chains. In fact, they're in every cell, right? They're carbon containing hydrogen, oxygen. They also contain definitely phosphorus and nitrogen. All right. So carbohydrates were carbon, hydrogen, oxygen. Lipids were carbon, hydrogen, oxygen, sometimes phosphorus. And proteins were the carbon, hydrogen, oxygen, nitrogen, and sometimes um, sulfur. But the nucleic acids are the Chopin, the Chopin with both the phosphorus and nitrogen. Okay, and no sulfur. But they're made up of what we call a nucleotide. All right, that's the monomer, kind of what makes a nucleic acid a nucleic acid. That nucleotide contains a nitrogen base, and it's going to be one of five types. You could have an adenine base, a thymine, a cytosine, a guanine, and then possibly a uracil instead of thymine if you're talking RNA. So those should be probably next to each other because, yeah, they're related, but they're different. Ribose sugar is another piece that we talked about of the deoxyribonucleic acid or RNA ribonucleic acid. So that's the ribose 5 carbon sugar, and then a phosphate group. Very rarely do you see phosphorus by itself. It's usually as a phosphate, that like PO4. Um, so DNA, stands for deoxyribonucleic acid, right? Let me pick a different color for that. Just means it's the ribose without the oxygen, one less oxygen than normal ribose. Cool. This is your genetic code. This is the core of eukaryotic cells, genetic code as well. Instructions for making the proteins, and as we studied, proteins just do everything else for the most part. For the most part. And they make your structure, and they make you work. Okay. It's also the basis for the molecule of ATP, which we'll talk about in a second too. So the bases, let me just say that the adenine, it's going to be double stranded in eukaryotes. So if it pairs, it's going to be adenine with the thymine, a double bond, or at least hydrogen bonds at two places, and then the cytosine at three places with the guanine. So yeah, they're at certain spots, kind of like a lock and key. The cytosine only likes to line up with the guanine, and the thymine and the adenine only like to line up with each other because of the orientation and the location of those um, hydrogen bonds, really. So yeah, we got sugar here, in this case, deoxyribose. And DNA is double stranded and has a certain kind of orientation. So one way it has a certain orientation and it exists exactly opposite direction with the other strand. So yeah, there's a certain symmetry to DNA. And it's held together by hydrogen bonds, which is how we're able to sort of unzip the DNA, sort of separate it, because you can interrupt those hydrogen bonds better than covalent bonds. It's found in the nucleus. For a while there, we did not know if it was the instructions for life. So we just knew they were acids. We called them nucleic acids because they were in the nucleus, in like this other section in the center of the cell. But it always stays in the nucleus unless the cell is replicating. And it replicates copies of itself in double. It's doubled. I should say. Is that what that I would say? So it doubles each time the cell divides. So all cells have all of your DNA, unless you're talking red blood cells, and then they just spat out. They used to have your DNA, but they just like spat out their nucleus. Isn't that weird? Okay, I guess they didn't need it. They're just kind of a place for oxygen to travel on. All right, so this is a picture of your DNA, double stranded, double helix is kind of what that ends up doing, just spiral. And then DNA is going to fold on itself. So you're going to have spirals of spirals, and you have proteins that kind of sort of maintain that sort of structure when it's time to separate the chromosomes, large DNA molecules. They have like folds on folds on folds on folds till you can probably see it in a microscope. Yeah, this macromolecule is so huge, so many tens of thousands of base pairs long that it's going to eventually be visible in a microscope. So you can kind of see the A's and the T's go together and the C's and the G's, and you can have one strand be code for a protein, and then the anti-strand like be the exact quote unquote opposite. Still possibly useful though. Could still maybe be used depending on the situation. Okay, so and that's the sugar phosphate backbone. We're saying we didn't draw it in there, but that's what the ribose and the phosphorus. Um, that's where that's located, but the nucleotides are in the middle. Okay, so RNA is not double-stranded. It's different by a couple things. One is that it just has a regular ribose, so we call it ribonucleic acid. Which is also needed for protein synthesis, but it's like the next step. Takes the instructions out of the nucleus so that we can make the proteins which are made outside of the nucleus. So proteins get built outside the nucleus. Since DNA doesn't leave, we need RNA for the message to take the message out. So we call it messenger RNA, and that's what gets the job done. Also, there's some other forms of RNA. So yeah, we call it mRNA when it's the messenger RNA. So the bases of, oh, by the way, yeah, M stands for messenger. Sorry. This time, instead of the thymine, it's uracil. So adenine does its possible double bond, or let's say if there was an adenine on the DNA in mRNA, we would have created a uracil. So it creates like an opposite copy of the DNA strand that it's getting the message off of. And then, of course, if there was a cytosine, there'd be a guanine in the mRNA, or if there's a guanine, then there would be on the DNA there would be a cytosine in the mRNA. Now, if there was a T, like there was a thymine, then it would have been an adenine on the mRNA. So we still add the adenine for the thymine. Yep, 5-carbon sugar ribose. Single stranded, yes, always. It does not like to be double stranded, and it's because of the uracil. It doesn't play nice with the others. Yeah, it doesn't allow for that hydrogen bonding. This is needed though. We need to do that because we need the single strand. Oh yeah, and it's a much shorter than the DNA. It's just a very small segment, so that we can form that protein that goes with that strand. So the mRNA, there it is. Messenger RNA copies the segment of the DNA, carries it out of the nucleus, and it carries it to a set of RNA molecules. Now, this is RNA that's functional RNA that actually acts as an enzyme, which it can. RNA can act as an enzyme. We call them ribosomes, by the way. Maybe you learned about it when you're learning about cells. And they're basically protein factories. They could be on the rough endoplasmic reticulum, which means ribosomal endoplasmic reticulum, or they could be in the cytosol. It's here that instructions are read to link the amino acids together in just the right way to build a protein. tRNA, that's what's supposed to go there. Transfer RNA, that's what that T stands for. It's made out of RNA, and it transports the amino acid to the 3-pair codon. It takes 3 base pairs, 3 nucleic acids to code for 1 amino acid. All right, and so if there's a mutation, there's a lot more combinations. There's 20 amino acids that are functional in your body, but there's a few combinations that could make that one amino acid. So if you did have a mutation for some reason, it may not ruin the protein structure, or it may still code for the same amino acid. The ribosome, by the way, we just call it rRNA. That's kind of lame. Ribosomal RNA, though. Yeah, actually forms a sandwich, and it helps read the messenger RNA codons and then transfer RNA. It's going to pick up amino acid, and we'll have that probably in like a video. We can see it happening, and it transfers amino acids. So then you're going to get that amino acid strand, and those are going to fold, and you're going to have wonderful things happen. So that's what, yeah, RNA can be a functional molecule like enzymes can. Another thing that is a nucleic acid is the ATP. Now, ATP is the energy molecule of the body, right? It's a form of energy that our cells can use to perform all their functions. Coincidentally, it takes an ATP for the transfer RNA to do its job and be able to bind to an amino acid and then find its way to the ribosomal strand at the ribosome and add its amino acid. That takes energy. So anything in the body takes energy, and most of the time it's in the form of an ATP. Yeah, we eat food so that we can make more ATP, which drives all the chemical reactions in your body for the most part. ATP stands for adenosine. That's that adenine base. And if it's charged, it's a triphosphate. That's what the TP stands for, not toilet paper. Okay, so yeah, an adenine nucleotide with the ribose sugar, of course. And this time it has 3 phosphate groups. The 3 P's are attached by high energy bonds. There's a lot of energy stored in that bond. When the bond breaks, usually it's just one, the energy gets transferred to the enzyme or to the tRNA or whatever is doing the work, and the cells will work. Maybe the protein channel has to be opened with an ATP, or protein pump, or channel. Yeah, that's like protein that like acts as a pump against the concentration gradient. We'll talk about that next chapter. So think of ATP as a rechargeable battery. Okay, there's its 3 phosphate groups. If it's charged, it has 3. Yep. And when it does its job, one phosphate group leaves. It's called phosphorylated. It like phosphorylates an enzyme. It like adds its phosphate group to an enzyme, which makes the enzyme kind of bend in conformational change. But remember, these phosphates are all a little negative. And it is going to do the work here, blah, blah, blah. And it's actually less charged. I think this is supposed to be smaller. I don't know. By one phosphate. And it changes from ATP, adenosine triphosphate, to ADP, adenosine diphosphate. And then it takes cellular respiration and a phosphate to get added back to recharge. So that's cellular respiration. And then we're going to have the 3 phosphate groups. So it's recharged. And ADP could be considered uncharged. There are times when ADP could donate another phosphate group, but it's a less high of an energy bond. So it's not as much energy. It doesn't want to do that. But yeah, you can make AMP, that's adenosine monophosphate, and a lot of times that's used in signaling in cells, signaling cascades, AMP. So yeah, nucleic acids used for ATP. The energy in the foods we eat are going to reattach that phosphate group. Otherwise, none of the chemical reactions in our body would be able to occur. Oh, yeah. So ADP then recharges to ATP. This is what we call phosphorylation. Anytime the phosphate group is added to something, we call it phosphorylation. Yeah, charged phosphate groups, when they break down, they can also release light, phosphorylation, but that doesn't really happen in our body. That's like separating the phosphorus. That's how phosphorus was discovered. vats of urine that were left to putrefy, right? And then as the bacteria broke down the phosphorus that was probably in the DNA, yeah, or the lipids, the phospholipid bilayer, then yeah, maybe it released some energy in the form of light. Yeah, phosphorylation requires a phosphate group, PO4, and it's taking place all the time in the mitochondria for recharging ATP. That's what's going to happen in the mitochondria. That's why the mitochondria is a powerhouse of a cell, right? It's like utilizing energy to recharge its batteries or recharge its debit card. It's more like a debit card that gets uncharged and then recharged. All right. So that's all we're going to cover for this class for DNA and nucleic acids, RNA, ATP. Here's the picture, by the way, of the adenine group with a ribose and 3 high energy bond phosphoruses that could be removed. Let me know if you have questions, and thanks a lot for joining us this evening. Glad that was a lot less drawn out and long as the as the one I did before. Yeah, those other ones, oh, that was a lot. Take care.